Really a pleasure to be here speaking with you today about a topic that I think is really very exciting. Exciting because it presents us with the first drug that really has extraordinary clinical efficacy in ALS, but also for what it teaches us about trial design and about the ALS condition and about how we can move forward with drugs to treat genetic ALS. So with that, I'm going to continue. And oops, okay, right. So here are my disclosures. I just want to mention that none of them really are terribly relevant for this talk. And I want to start not by giving us a timeline of ALS, but by giving us a timeline of development of the drug tofersen. And I'm going to touch on some of these points in more detail and just sort of mention them with this timeline. As all ALS timelines do, and frankly, as all neurology timelines do, even in the US, they do begin with old white European men. So that is just something in general for ALS. This happens to be a French neurologist, Jean-Martin Charcot. He is often considered one of the fathers of neurology. ALS was originally called La Maladie de Charcot. He described it back in 1869, and the condition bore his name for quite some time. He's also, by the way, famous for many other things. One of the things he's most well known for actually are studies on hysteria, the wandering uterus, as we now call it. Nothing really happened in the field of ALS until something in the US. And I don't know how often you guys across the pond talk about Lou Gehrig, but in the US, we talk about Lou Gehrig a lot in relation to ALS, and we still often call it Lou Gehrig's disease. Because in 1939, one of our greatest athletes, unfortunately, was diagnosed with ALS, and he subsequently passed in 1941. But before he did, he gave one of the greatest speeches known to man, where he called himself the luckiest man. And if you haven't had the pleasure of hearing that speech, I recommend that you search for it online and listen to it because it's really quite moving and quite wonderful. Again, some time passed, and then some really important things start to happen. So in 1993, just before my time in the field, when I don't even think Dr. Turner was around yet, but and I'll give him credit for not being around yet, the SOD1 gene was discovered. And at that time, I'm told that people who were in the field thought we had this thing licked. The cure was around the corner. Now we are going to, be able to find treatments and find, just get this thing under control. And there was a ton of excitement in the field. As we know, that was maybe a little bit too much excitement, but it did eventually lead to some very great movements forward. But unfortunately, some time did pass until now we're getting to modern times. In 2020, the phase 1/2 data for tofersen was published. And if you don't recognize the picture there, you should, because I'm told that in Europe, this is a very, very popular sport, biathlon. In my home, we actually like it too. And what I'm picturing here is the athletes shooting at their targets. And the most important thing, and we'll discuss this, about the phase 1/2 data for tofersen is that it showed target engagement, which I understand is extraordinarily important in biathlon. So this is when things really start moving. In 2021, EAPs, which are early access programs, became available in the UK and the USA for tofersen. And a study called the ATLAS study began in the US. And the ATLAS study is a study of treating pre-symptomatic individuals who carry the SOD1 gene with tofersen. In 2022, the phase 3 data for the VALOR study was published. And again, we're going to go over this in some more detail. This is just a timeline. In 2023, the FDA, which is our version of your EMA, gave accelerated approval, and that's an important word, accelerated approval for tofersen on the basis of biomarker data, which I will go over. In 2024, your EMA gave scientific recommendation for the use of tofersen in the EU and the European Economic Area. And then in 2025, the Medicine and Healthcare Products Regulatory Agency granted marketing authorization in the UK, although I do understand that this is different from getting payers to pay for it. And then finally, the last timeline of notice is the Valor open label extension data was published just very recently in 2025, which is the most exciting data we've probably ever seen in the ALS field. So we're going to talk in some detail about this so we can all have an understanding of the progression of how we've come to understand this data and what it's taught us about the disease of ALS, how to do trials, and how to move forward also in the genetic field of ALS. So just to level set, because I think this is important, I wanted to just briefly go over the mechanism of the drug tofersen. So as you probably know, I'm not sure if you can see my pointer, but as you probably know, SOD1 ALS is what we call a toxic gain of function mutation. And what that means is that the mutation in SOD1 causes eventually down the line production of a protein, the SOD1 protein, that is toxic and does damage. We're not going to go into how it does damage. That's not entirely understood, but the point of the matter is that the mutation in the gene, which is DNA, causes transcription of the DNA into mRNA, and that mRNA is then produces the mutant protein. So the drug tofersen is a synthetic piece of DNA. This is the synthetic piece of DNA. That synthetic piece of DNA is taken up into the cell by endocytosis. That's just a process of how cells take up material, and in this case, it's the drug tofersen. That piece of DNA can bind complementary to the mRNA, to the mutant mRNA, and then the drug takes advantage of a protein, a naturally occurring protein in the cell called RNase H. RNase H is a protein that, as I said, is naturally occurring in the cell. And the job of RNase H, which is the full name for that is ribonuclease H, if you wanted to know. This is an enzyme that breaks down combinations of DNA and RNA, because technically speaking, combinations of DNA and RNA shouldn't really exist. So this RNase H binds to this combination of the synthetic DNA, which is tofersen, and the mutant mRNA, and breaks it down. And since this then mutant mRNA is taken out of the environment, it can no longer produce the SOD1 protein, and so you have less mutant SOD1 protein, and that's where the magic is. So that's how the drug works. And it's really quite remarkably simple, actually, and quite brilliant, if you ask me. So that's how the magic works. And then, so moving on to the first trial that I mentioned already was the phase 1/2 trial. So this trial was a very short trial. It was a 12-week trial, which, you know, in the time course of ALS is just a blip. And this is what we call a MAD trial, a multiple ascending dose trial. And all early phase trials, as you may know, the primary outcome is safety. So we just really want to make sure that nobody gets hurt from this drug. And we also want to start looking at what the maximum tolerated dose is. So how high up can we push the dose and keep it safe? But we also like to look at early efficacy, and we like to see something that is demonstrated here in this graph, which is something called a dose response curve. And what that means is that the people in this trial who got the highest dose, which is represented by the orange line, had the highest what we call target engagement, if you remember my little biathletes from the timeline. So what's represented here on the y-axis is the concentration of SOD1 protein in the CSF. So these people very generously submitted themselves to multiple lumbar punctures, and the SOD1 protein was measured in the CSF. And those who got the highest dose of drug had the lowest amount of SOD1 protein in their CSF. And since we've agreed that SOD1 protein is bad, the lowest amount would be a good thing. So that was very exciting because it essentially showed target engagement. The drug is doing what we hoped it would do. And there were very few safety signals, meaning that the drug was well tolerated. So for a phase 1/2 trial, this is really a very, very positive result. But the gravy in this trial was this early evidence of potential clinical efficacy. So in a phase 1/2 trial, you don't need to see clinical efficacy, but when you do see a hint of clinical efficacy, it really gets your blood running. So these are called secondary endpoints because it's not necessary to see this to have a successful early phase trial, but it's very, very nice. And the things they measured are the usual and customary things that we measure in an ALS clinical trial. So the ALS functional rating scale, the breathing test, which is the slow vital capacity, and handheld dynamometry, which is strength testing with a little device that quantifies muscle strength. And what you can see here is that the treatment group is in, the treatment group is in orange and the placebo group is in blue. And higher numbers are better because you want a higher functional rating scale. You want a higher score on the breathing test, and you want higher scores on your strength testing. And generally speaking, the treatment group looked a little bit better than the placebo group. None of this was statistically significant. As you can see, the numbers here are very small. So there were 12 people in the placebo group, 10 people in the tofersen group. These lower graphs down here, the individuals were divided into groups that were predicted to be faster progressors versus non-fast progressors, and that was based on specific mutation in the SOD1 gene. These groups are extremely small, just 4 people, and I think we should really never make any conclusions when we're talking about groups that small, even if it's tempting. So those were the very early results of tofersen, and they were really, really very exciting. So we wait, and it was decided, of course, on the basis of that early phase trial that a phase 3 trial would be undertaken. The phase 3 trial was called Valor. At Penn, we were very pleased to be a site for that trial. It was very exciting. That was a relatively large trial. That trial was 108 individuals. It was randomized 2-to-1, such that 2/3 of the individuals were given active drug and 1/3 were given placebo. Instead of a 12-week trial, that was a 6-week trial. And then, I'm sorry, a 6-month trial, excuse me. After 6 months, all of the individuals in the placebo group were transitioned to open label extension. The primary endpoint in this trial was no longer a biomarker, but was the ALS functional rating scale. And we waited for those results. 6 months doesn't sound like a long time, but it is in the ALS world for individuals with ALS, for family members, for the ALS community, and for researchers alike. So this was a very strange occurrence, and I honestly do not remember the circumstances under which it happened, but I was not at the meeting where the results were presented, but for reasons that I never totally understood, the meeting was televised, and you could actually watch the the presentation of the results. And the person who was giving the talk was a very well respected person in our field named Dr. Tim Miller. He was the PI on this trial, and he's an extraordinary presenter, and he presents in a very even tone. If any of you have had the pleasure of hearing him present, he's very clearly spoken. He gives away nothing in his presentation, but I was literally sitting on my couch getting ready and I to hear this because I was expecting really good news. And I told my family that they're about to see history because we're about to see the first really successful drug for ALS. And I was sitting on my couch, all excited to hear the presentation. And this is what we saw. So I looked at this quickly and I thought, oh my God, it's a miracle. It works, because I thought I was comparing the blue lines to the red lines, which are clearly different. And then I looked down at the p-values, which you see here. And I was listening to Dr. Miller's voice, which is, if you've ever heard it, it's very even toned. And I realized that, oh my gosh, we missed our primary endpoint, because it wasn't the blue lines compared to the red lines. It was the solid blue compared to the dashed blue, and the solid red compared to the dashed red. And in fact, there was no statistical significantly difference between the ALS functional rating scale um, and the primary endpoint, in fact, had been missed. And I was shocked. And I remember sucking wind, and I couldn't really um, sort of understand what was going on around me for just a moment. And then the next slide came up, and this was the next slide. And here we see the biomarker, the SOD1 protein. And you can see very clearly now the dashed lines and the solid lines clearly are separated. And these p-values are very significant. So what does this tell us? Well, this tells us that the biomarkers were very significantly different. And now we've shown over a longer period of time that, in fact, there definitely is target engagement. And so I breathed a sigh of relief. And I don't have this slide here, but the results were very similar for neurofilament. And neurofilament, of course, is a more general biomarker in ALS, not specific to SOD1 disease, but neurofilament is a marker of axonal degeneration, and axonal degeneration in general is bad. And so when neurofilament goes down, that is generally a good thing because it means less nerve cell death. And that was essentially the same as the SOD1. So how do we interpret this data? What do we do with this information that we didn't find a clinical outcome, but we found a very impressive target engagement outcome that we think is very meaningful in SOD1 disease? So it's time to circle the wagons. How do we think about this? What do we do? Do we jettison this whole program? Do we say, well, the surgery was successful, but the patient still didn't do well? I mean, how do we interpret this? So what Biogen did, which I think was very well, clearly in retrospect, it was very smart, is they just waited. They just waited to see what would happen with more time on the drug. And given that there were no really problematic safety signals with the drug, which is to say that it was well tolerated, there were a few serious adverse events, but none that were catastrophic. And in general, the drug was considered safe. So we waited, which again, for the ALS community, for patients, for family members, for loved ones, for physicians is difficult, but sometimes we do difficult things. So at 52 weeks of data, this is what we saw. And now what does this tell us? So this is more biomarker data. So now we have data on SOD1 protein and neurofilament at 52 weeks. The light blue line is at 28 weeks, which is approximately, which you can see there, is approximately when the individuals who were originally in the placebo group start to get the drug. So they transition to the open label extension. Now, this, if you haven't looked at data like this is something worth taking a minute to understand, because this is where it all starts coming together. So the group that's called Valor is the group that was in the original treatment group, and they start getting drug at time 0. The group that's called OLE is the original placebo group, and they start getting drug in the light blue shaded area. So the OLE group is dashed when and open circled, and they're dashed when they're getting placebo, and they become a solid line when they're getting drug. So they're the blue people. The Valor people are the people who got drug all the way through. So what I want you to look at, so first we're going to look at the CSF SOD1 protein graph. And what I want you to look at, first we're going to look at the green line. So these are the people who got drug from the very beginning. And it looks like the SOD1 protein really hits near bottom around 8 to 12 weeks. And it stays there. So the CSF SOD1 protein naders or hits bottom around 8 to 12 weeks and stays there. So oops, I'm sorry. How do I go back? There we go. So we've hit bottom at 8 to 12 weeks and we stay there. Now for the people in the Valor group, their CSF protein bounces around, CSF SOD1 protein bounces around a little bit, but then they get drug here, and their CSF SOD1 protein is still all the way up here, which is about where they started, right? Then they get drug, and by 40 weeks, which is roughly 8 to 12 weeks after they get drug, their CSF SOD1 protein also naders. And by 52 weeks, there's still no difference between the early start and late start group. The reason that we say there's no difference here is because these error bars overlap. So these two numbers are not statistically different by 52 weeks. But what we can confidently say is that the individuals who got drug first, they hit their nadir much earlier than the individuals who were not on drug. Now, if we transition over to the neurofilament graph, we see essentially the same thing, just a little bit delayed, because neurofilament drops after CSF SOD1 protein. So neurofilament naders around 12 weeks. So looking at the individuals who started getting drug at time 0, their neurofilament starts bottoming out very quickly, and they hit their nadir somewhere around here-ish. Maybe it keeps going down a little bit, but it's definitely down here. The individuals who are not getting drug, their neurofilament stays elevated, then they get drug, and it clearly starts going down. And by 52 weeks, there's definitely no difference between the CSF neurofilament between the two groups. And that's what this data shows. Okay, so looking at some clinical endpoints in the open label extension. And again, this is 1 year on drug. So now, again, we're looking at the green people to start with. These are the people who get drug at time 0. Now, people with ALS get worse. So these lines we don't want to be going down, but unfortunately they are because the ALS-FRS R score tends to go down. So they get drug here, the green people, and their ALS-FRS-R goes down. What you might imagine, although you have to imagine it a little bit to be honest, is that around 28 weeks, perhaps the slope of this line levels off a little bit. Now, if that were true, around 28 weeks, you can see these individuals are still progressing because they get drug here. And by 52 weeks, they haven't actually hit 28 weeks. Perhaps the slope of their line is leveling off a little bit here, but it would really be too early to say, and we probably shouldn't. Um, so with regard to HHD, which is handheld dynamometry, the individuals who got drug starting here, they're getting weaker, perhaps at 28 weeks. If you want to be an optimist, you might say, are they getting a little bit stronger? Certainly, it's not statistically significant. Are you someone who wants to look at trends in the data or not? That's up to you. These individuals here who aren't getting drug, they're certainly getting weaker. They continue to get weaker as time passes. Is there any chance they're leveling off? Again, definitely not statistically significant, but this is the data that we have to look at. Okay, so from our first open label extension, these are our conclusions. By about 8 weeks, tofersen reduces SOD1 protein, showing target engagement. By about 12 weeks, tofersen reduces neurofilament, which indicates a reduction in axonal injury and neurodegeneration. By 28 weeks, trends suggest a slowing of clinical decline, again, not statistically significant, but perhaps a trend towards clinical improvement. So by 52 weeks at the open label extension, earlier initiation of tofersen has potentially reduced decline in clinical function, et cetera. And the data at that point showed a 30% reduction in mortality, which the endpoint included permanent ventilation. I should say permanent invasive ventilation. So moving on to the newest data. So remember I said we had circled the wagons. And this is what circling the wagons gave us. So Biogen continued the study, even though the drug was approved by this point in the US. So the drug is approved, and it was approved, by the way, I should mention, on the biomarker of neurofilament. Because remember that VALOR, the phase 3 study, failed to hit its primary clinical endpoint. So the drug could not be approved on clinical findings. And the FDA in the US and the EMA in the EU did grant conditional approval on the basis of using neurofilament as a biomarker, which was quite a milestone. And I'm going to address that shortly. But just to finish out a discussion of our data, so now, just recently, we had publication of the data at 148 weeks. Now, 148 weeks is just shy of 3 years, which again, in the world of ALS is an absolute eternity. And I don't want to look past that. But what we're showing here is importantly the difference between getting drug earlier and getting drug later. Now, I'm going to start with looking at the biomarker. And this time I'm not showing you SOD1, I'm showing you neurofilament. Because again, neurofilament is a measure of neurodegeneration. And here, our colors have changed, apologies. Here, early start tofersen is orange, and placebo or delayed start tofersen is in black. And the individuals who started tofersen at time 0, what we see is that by 12 weeks here, neurofilament has gone down. It hasn't necessarily natered, but it's gone down, and it stays down. And for the late start individuals who are here in black, they start their drug here. So if 28 plus 12 is 40, So their neurofilament goes down at 40 weeks and stays down. And by, certainly by here, we have all overlap of, by 52 weeks, we have all overlap of neurofilament, and there's no statistical difference between the two. With regard to the clinical results, so these are the results for the ALS functional rating scale. What you see here is, again, the orange people are early start. So they start getting their drug here. And at 28 weeks, if you want to imagine that the orange people are sort of, so here's the slope of the decline in the functional rating scale for the first 28 weeks. Oops. And sorry, if you want to imagine that the slope is leveling off, you could perhaps imagine that. And then you have your later start people, and this is the slope of their decline. They get the drug at 28 weeks. So then by around 56 weeks, which would be somewhere in here, you could imagine perhaps that the slope is leveling off. And certainly by here, you could say that the slope has leveled off. But the important thing to notice is that at around 124 weeks onward, these error bars are not overlapping, such that there is a difference all the way out here between the two groups, and the group that got drug earlier is doing better than the group that got drug later, and that difference persists over the long term. And what that tells us, which should not come as a surprise, is that early treatment matters. And we've always postulated that in a neurodegenerative disease, the earlier you treat, the better. The earlier you lower SOD1, the better. The earlier you lower neurofilament, the better. And this is proof in the pudding of that. And this is the first time that we ever used a model like this, looking at early treatment versus late treatment. And this is really the first time also that one has ever shown that ALS might be a treatable disorder. And when I say treatable, I mean truly modifiable in the sense of slowing disease progression. Because if we really are flattening out the curve here, and you can decide if you believe it, I do. I really feel that this slope here is quite different from this slope here, and it is not consistent with natural history. But the slide I'm going to show you next is the one that really convinced me. It's nice to look at graphs, but the slide I'm going to show you next is more, oh no, actually, the one after this. Sorry if I got you excited. It's just because I'm excited. So these were the other secondary endpoints. And this one is the breathing test, the slow vital capacity. And what you can see here, again, the orange folks who were treated early, this is their decline. And then it seems to start to level off a bit after 28 weeks or so. These are the later start people. They get treated here. And, perhaps you can imagine that things are starting to level off somewhere around here. But we cannot claim that there's a difference here. But the reality of that is that perhaps there's a little bit of improvement here, although to be honest, I think that's a tough sell. And here with the handheld dynamometry megascore, there's a clear decline here. And I think to be fair, this is also a tougher sell in terms of the statistics because there's a lot of bouncing around here. And I'm not sure if we drew a straight line through here, how different the slope would actually be. So I think that this is a bit of a messier statistic that needs probably a little bit more data to figure out. And I probably wouldn't hang my hat on this one per se. But what I would hang my hat on is the next slide, which is the one that I find really the most exciting. And that's this one. So this here is a different way of looking at the data. So this slide shows individuals who are divided up by neurofilament when they started the study. So what we do know is that neurofilament predicts survival. So high neurofilament is not a good thing because it in general says how much neurodegeneration is going on. So if you stagger individuals by neurofilament, you can, to some extent, predict prognosis. Now, if you divide by faster progressing and slower progressing on the basis of neurofilament, and then based on what group they were in, original treatment versus placebo, and then you look at what happened to them with respect to their functional rating scale, their percent predicted SVC, and their handheld dynamometry megascore. There were in every single one of these groups, there was a percentage of people who improved or stabilized. And so if this were a waterfall plot, if any of you are familiar with what a waterfall, whoops, sorry, if any of you are familiar with what a waterfall plot looks like, a waterfall plot plots the data of every single person in the study. And then you can visually see how many people fall below the line, meaning that they didn't do well, versus how many people are on the line, meaning that they stayed steady, versus how many people are above the line, meaning that they improved. So for example, 12.5% of the people in the fast progressing subgroup who were originally in the placebo group but eventually got drug improved versus 16% of the people who were in the fast progressing subgroup but and originally got drug improved. The point is that we have never in an ALS study seen anyone improve. So the likelihood of this happening by chance is zero. So they modeled this out. So I'm not just saying it's zero, it is actually zero. That we would have found this by chance, saying that this wasn't a drug effect. So there is no chance that this outcome was not related to the drug. The reality is that a single drug isn't going to work for everyone. In the world of hypertension, for example, we have multiple different categories of drugs because not one drug works for every person with hypertension. So you can't expect to see 100% in all of these boxes. The fact that we see non-zero numbers in all of these boxes is absolutely remarkable. And that's why I am a firm believer in this drug, a firm believer. We have to talk about safety profile because you can't give a talk like this and not. The point is this is a safe drug. There are very few side effects. We have lots of clinical experience with this drug. The only basically serious side effect was myelitis, which is an inflammatory reaction in the CSF that caused one person to stop the drug. It did not cause harm to the person. It just means that this person had an inflammatory reaction in the CSF and could not continue the drug. Otherwise, it's very well tolerated. The take-home points that are learnings from tofersen, which is really the most important thing because this drug, obviously, this is not a one-size-fits-all drug. So not everybody with genetic ALS can take it. It doesn't work for sporadic ALS, obviously, but we've learned really important things from going through this whole exercise. And what we've learned is that neurofilament is a good biomarker for ALS, and it can be used in other ALS clinical trials for go, no-go decisions, and the FDA at least will consider it for accelerated approval decisions. This concept of using early treat versus late start to prove that early treatment matters and to allow us to look at long-term data with open label extension is extraordinarily valuable. It's a little bit complicated. I've tried to explain it to people in other fields, in other neurologic fields, as a good way to do studies and to do them in a more expedited fashion. And not everybody gets it. So if it's feeling a little fuzzy in your head, that's okay. Sit with it a little bit and maybe it'll take hold. As I tried to indicate, this is the first ever evidence that ALS is a truly treatable disease, which is monumentally exciting, I think. And also, as I mentioned a little bit early in this talk, this paves the way for treating of pre-symptomatic individuals who carry a gene for ALS, which is really the holy grail, and which is what we're all hoping for one day in the very, very near future. And I think that this is extraordinarily exciting. And this is going to be the first of the dominoes to fall in genetic ALS. And now that we know how to do a trial like this, when the science catches up a little bit with some of the other mutations, we will have a clear line of sight as to how to design a trial, and there won't be as many little hiccups, and we have a lot to be looking forward to. So, oh, and I want to end this talk where I started with a quote from the great Professor Charcot. He said back in the 1800s, if you do not have a proven treatment for certain illnesses, bid your time, do what you can, but do not harm your patients. And I'm looking very forward to the time when we don't have to think like that at all for ALS. And we can really hand out treatments that are disease modifying and not just do no harm.